In this video lecture, I will be talking on recombination activating genes RAG1 and RAG2. The recombination activating genes encode proteins that play crucial role in recombination of gene segments that encode TCR, as can be seen here, and antibodies. And this enzyme, which is produced by these genes, are critical for generating diversity of TCR and antibodies. So RAG1 and RAG2, they are expressed by or in developing B anti-lymphocytes. As can be seen here, this is RAG1. The function of RAG1 is recognition. Actually, RAG1, it recognizes the spacer of recombination signal sequence. But however, the stability of RAG1 require binding of RAG2 to form a protein complex. And as can be seen here, RAG enzymes bind to recombination signal sequence that flank the variable region and the joining segment of the TCR alpha, and they flank this recombination signal sequence flanks the variable of beta TCR and the joining beta TCR. This is on the 5 side, and this it flanks the variable beta on the 3 side. But however, they flank the diversity segment or this segment on both sides, as can be seen here. TCR should be able to recognize around 10 to power 16 different pathogens. But however, TCR DNA, these are the TCR, these are the DNA encode TCR alpha, and these are the DNA segments encode TCR beta. Has fewer variable, as can be seen here, has fewer variable gene segments. These around 70 variable gene segments in TCR alpha, in TCR beta around 50 or more, a little bit, and these variable segments, they are around 3,640 gene pairs, and however, to face around 10 to power 16 different pathogens, so some mechanisms or possible mechanisms are required, and these possible mechanisms, they contribute and generate a great diversity or a high degree diversity of the TCR. So these mechanisms, for example, random gene segments recombination and junctional diversity in form of B nucleotide and N nucleotide addition. The binding of the segments or gene segments that encode TCR alpha and TCR beta are directed by the recombination signal sequence according to the role 12 base pair to 23 base pair. As can be seen here, this here, the joining is flanked by 12 base pair and the variable flanked by 23. So the direction of binding, it will be to this direction from 12 to 23. And since RAG complex has the capacity to recognize the spacer of 12 base pair and spacer of 23 base pair, so the recombination is initiated by recombinase enzyme or by the RAG complex proteins. First, this process starts by recognition. As I mentioned, that RAG1 has the capacity to recognize 12 base pair and 23 base pair spacer of recombination signal sequences, and this is followed by the orientation of the variable and joining, like in this position. This actually directed by the RAG. Then this is followed by the cleavage of the space, as here the RAG it cleaves the space between the coding segment and the recombination signal sequences. This cleavage actually it leads to formation of hairpin, which is associated by adding B nucleotides. As can be seen here, this is a variable region and this is a joining segment. So the RAG will add nucleotides. These are templated nucleotides as a coding site, and this side also, this coding site. And as can be seen here, this is TCGA. So on the opposite direction, you can read them TCGA, the same. So this is templated or B palindromic nucleotides or B nucleotides. However, the gene now is open and cannot be left open. RAG again, it will cleave the hairpin, and this area it will become like this. It is now this is the hairpin cleaved. So another enzyme known as terminal deoxynucleotide transferase, together with other enzymes, for example Artemis and DNA ligase, but however the main is done by the terminal deoxynucleotide transferase. The terminal deoxynucleotide transferase, it adds some nucleotides just to repair this open area of the DNA. But however, this is a non-templated. This is in case of TCR alpha, and in case of TCR beta, it looks like this, and actually this will result in diverse TCR and alpha and TCR beta, which could reach to 10 to power 16 different TCR segments, and this will face the number of the expected pathogens in the milieu, which is around also 10 to power 16. So RAG1 and RAG2 RAG2, they play very crucial role in the diversity of the T cell receptor. Thank you very much, Professor Ahmed Boulad.